Hello everyone, welcome back to my channel. In this video lecture, we will cover our first chapter of class 11 biology, that is plant growth and development. So, development is nothing but growth plus differentiation. Growth is irreversible, permanent increase in size of an organ or its part or even of an individual cell. It is accompanied by metabolic process. Example, expansion of leaf growth. Growth is measurable. Growth may be considered as an increase in amount of protoplasm, can be measured in terms of increase in fresh weight, dry weight, etc. Single maize root apical gives rise to more than 17,500 cells per hour, and whereas watermelon cell may increase in size by up to 350,000 times. Growth in pollen tube is measured by length, and in dicot leaf it is measured by surface area. Shoot apical meristem is the primary growth longates of axis, and dicot and gymnosperms, lateral meristems, vascular cambium appear later in life. Plant growth is generally intermediate and unlimited growth in plants due to presence of meristems. New cells are added always, hence it is termed as open form of growth. Phases of growth: meristematic phases, elongation and maturation phase attains maximum size, wall thickning, protoplasmic modification, most of the tissues and cell type. Growth rates: increases growth per time is termed as growth rate. It is defined into two: arithmetic or geometric growth. Arithmetic growth follows mitotic cell division and only one cell continues to divide while other differentiates and matures. Linear curve is observed on LT graph, whereas in geometric growth, initial growth is slow and it increases rapidly thereafter at an exponential rate. Here, both progeny cells following mitotic cell division retain the ability to divide and continue to do so. And here, sigmoid curve is obtained. Quantitative comparison between two living systems. Absolute growth rate is the total growth per unit time comparison, whereas in relative growth rate, growth per unit time expressed on common basis. Condition for growth: water, cell enlargement, turgidity, O2, nutrients and optimum temperature are important, and any deviation from optimum temperature is the determinant to its survival. Growth and further development linked to water status. Now, what is differentiation, dedifferentiation and redifferentiation? Cells of meristems differentiate to form mature cell. Mature cell, it is called differentiation. Example, to form trachyary element, protoplasm is lost. Strong elastic secondary walls develop to handle extreme condition. Differentiated cell regain capacity to divide is called dedifferentiation. Example, formation of meristems. Dedifferentiated cell again lose capacity to divide is called redifferentiated. Growth in plants be intermediate or determinate or differentiated is always open. Now, what is development? It includes all the changes that an organism goes through during the life cycle. Plasticity: plants follow different pathway in response to environment or phase of life to form different kinds of structure. Example, heterophyll in cotton, coriander and larkspur. Heterophyll due to environment in buttercup. Plant growth regulators. First, characteristics. They could be indole compounds, adenine derivative or carotenoid derivative, or terpene. PGR may be growth promoter, example auxin, gibberellin, cytokinin, or may be growth inhibitor, example abscisic acid or ethylene. Second, the discovery of plant growth regulator. Auxin, Charles Darwin and Francis Darwin. So photoprism in coleoptile or canary grasses. F.W. Wendt isolated auxine from coleoptile of oat seedlings, site of transmittable influence. Gibberellin Bacon disease of rice seedling was caused by fungal pathogen, and E. Karsava in 1926 detected that it was gibberellic acid substance. Symptoms appear when treated with sterile filtrate of fungi. Cytokine F. Skou found that the cell division in tobacco internode takes place only if the auxin extraction of vascular tissue, yeast, coconut milk, or DNA is added. Callus is the mass of undifferentiated cells. Abscisic acid ABA. During mid-1960s, 3 researchers discovered inhibitors B, abscission 2, and dormin, which were collectively named as abscisic acid. Fourth, ethylene. Cousins confirmed release of volatile substance in ripened orange that ripens the banana, which was later known as ethylene. Third, physiological effects of plant growth regulators. Auxine, first isolated from human urine. Then these are used in agriculture and horticultural products and practices, used for plant propagation, promote flowering, used in tea plantation, cause apical dominance, and to overcome precipitation it is done, used as herbicides. Whereas gibberellins, there are hundreds of gibberellins, out of which GA3 is most studies. All are acidic, increased in length of grape stalk, improve shape of apple, delay synthesis, speed up malting and brewing, hastens maturity period in juvenile conifers. Cytokines, discovered as purine, that is kinetin, from autoclaved heron sperm, DNA which does not occur naturally in plants. Naturally it is found as zeatin in corn, found in rapidly dividing cell zone, helps to produce new leaves, chloroplast lateral shoot, adventitious roots, overcome apical dominance, and delay of cell senescence. Ethylene, synthesized by tissue undergoing senescence and ripening fruits, horizontal growth of seedlings, swelling of axis, then promotes abscission of leaves, flowers, then increase respiratory rate, breaks seed dormancy, initiate germination in peanut seed, and many more. Abscisic acids regulate abscission and dormancy, inhibit seed germination, stimulates closure of stomata. By inducing dormancy, it helps seed to withstand desiccation. Then what is photoperiodism? Response of plant to period of day-night is termed as photoperiodism. It is very important topic. Duration of dark holds same importance as night. To detect the light, leaves are used on a hormone is sent at the site of flowering. Now, vernalization, it prevents precocious reproductive development. Flowering either quantitatively or qualitatively depends on low temperature. It enables plants to have sufficient time to reach maturity. Wheat, barley have 2 varieties. If winter variety is grown in spring, then it may fail to flower. Hence, they are planted in autumn. But spring variety is better. Winter variety is harvested in midsummer. Another example, monocarpic plants that normally flower and die in the secondary season. Example, cabbage, carrot, sugar beet, etc. Now, what is seed dormancy? It is under control of endogenous conditions. Reasons are impermeable hard seed coat or presence of chemical inhibitors or immature embryo. How to overcome seed dormancy? By mechanical abrasions using knife, seed sandpaper, etc. or vigorous shaking. Naturally, these abrasions are provided by microbial action, passage through digestive tract, or by chilling or treating with GA or nitrates. Changing environmental conditions such as light and temperature. So this is the end of the chapter. Hope you liked it and understood. So please like and share my videos. Thank you for watching and don't forget to subscribe my channel. Thank you. Till then, bye bye.